I have to record. All right, so metabolism, it's comprised of two. Oh, before I do that, let me go back to the previous PowerPoint. Right here, go back to the definition of metabolism, a collection of all these reactions, chemical reactions in the cell. Ultimate goals of metabolism: growth, repair, and reproduction. So you should remember from biology 1406, there are two types of metabolic reactions, catabolic and anabolic. Catabolic reactions are the ones that break things up, and in the process, they are going to release energy. So these are exergonic reactions because energy is being released by breaking chemical bonds. Okay, these are hydrolytic exergonic reactions, and these are the ones that make ATP. On the other hand, anabolic reactions are the ones that build, make things. These are the ones that make molecules for the cell. They're used for repair and growth. So these are the ones that make cell walls, cell membranes, proteins, replicate DNA. So anabolic reactions are the ones that make things. However, anabolic reactions need energy. They're endergonic reactions. So that's how these two types of reactions are related to each other. Anabolic reactions are going to provide the energy that, catabolic reactions provide the energy that anabolic reactions need. And this is the picture that tries to explain the relationship between catabolism and anabolism. And so catabolic reactions are going to take in molecules like glucose, and they're going to break them up. And in the process, two things are released. First, energy is released. Okay, we already talked about that. Energy is being released, but also, almost as important or more importantly, subunits, raw materials are being produced. So this produces raw materials. Okay, raw materials that anabolic reactions are going to use together with the energy to build things up. So remember, for example, if a bacteria is making cell walls, the cell wall is made of a peptidoglycan. The glycan part are essentially glucose molecules that have been modified. So catabolism would provide the cell with the glucose that the cell can use to modify with the energy that catabolic reactions produce to create these glycan chains, for example. So don't forget that the raw materials are also a product of catabolism, not just the energy. And that anabolism needs two things. It needs the raw materials, the subunits, the building blocks, the amino acids, the monosaccharides, the nucleotides to build cellular molecules, cellular components, obviously also energy. All right. So we talked about these already. Know which reaction is endergonic, which one is exergonic. And this just goes through a list of the things that metabolic pathways need. Enzymes, ATP, chemical energy, electron carriers, etc. So we're going to go through all these components of metabolic pathways. And we have already mentioned energy, ATP. So ATP is we call it the energy currency of cells. Remember that ATP from chapter 2 is a nucleotide. It comes from a nucleotide that has been given two more phosphates. It is an adenine nucleotide. And it is the most common energy currency of cells, period, just all cells, not just bacteria. All right, so there's the structure of ATP. Again, it's just a nucleotide, is an adenosine triphosphate, and the last two phosphates are surrounded by negative charges, which make them very unstable, especially that last one, so that these bonds are easily broken. So ATP is made from ADP, and actually ADP is made from AMP, adenosine monophosphate. So that's what makes ADP. So that's a separate type of reaction. But to make ATP, the cell is going to take ADP, add one more phosphate to it. This is going to require energy because you're adding negative charges to negative charges, and they're supposed to repel each other. So you're going to have to force them together. You can see this squiggly line saying these are very unstable bonds, and that's what makes ATP have high energy bonds. ATP is then hydrolyzed. When the last phosphate is removed, energy is released, and now we're back to having ADP. So to make ATP, we're going to need two things: energy, and we're going to need phosphorus. So the process of making ATP is called phosphorylation. There are three mechanisms of phosphorylation. One of them we don't care about, which is the one that photosynthetic organisms use. So we're not going to talk about that one. We're going to talk about the types of phosphorylation that non-photosynthetic organisms use. So there's two of those: substrate level phosphorylation and oxidative phosphorylation. Okay, substrate level phosphorylation is going to use chemical energy to add a phosphate to an ADP that is released in an exergonic reaction. Okay, so this is going to occur in the middle of a chemical pathway. So this will be one more step in a chemical pathway, substrate level phosphorylation. Oxidative phosphorylation, on the other hand, is a much more specific event in that there is an enzyme whose sole job is to make ATP. Okay, so let me give you a little bit more of an explanation on both. This is an example of substrate level phosphorylation, making ATP by using the substrates of an enzyme. Okay, so again, with substrate level phosphorylation, the process of making ATP is just one step in a chemical reaction, in a series of chemical pathways, in a pathway that has many steps. And this making ATP step is just one of the steps. So there are many of these substrate level phosphorylation pathways. One example is one in which a molecule, which is one of the last molecules in glycolysis, called PP, phosphoenol pyruvate, which has a phosphate attached to it. There's an enzyme that is going to remove that phosphate. And just to note here, notice that the phosphate is surrounded by chemical groups, which are, again, unstable and therefore promote a chemical reaction. So that phosphate is surrounded by this ketone group here and a double bonded carbon here. So this is actually a phosphate that can be removed because it's surrounded by chemical groups. So the enzyme is going to remove the phosphate and attach it to ADP, producing ATP, and the PEP is now turned into pyruvate. So it's just a matter of removing the phosphate and giving it to ADP, and that's a process that happens by the action of this enzyme. Okay, so let me just point something out here. The enzyme right here is not a specific enzyme, is not the only enzyme that does these. Any enzyme capable of removing a phosphate out of a chemical compound in which the phosphate is surrounded by unstable groups, chemical groups, reactive groups, will do the same thing. Okay, so there are many enzymes that can do this job. All right. On the other hand, oxidative phosphorylation is a much more specific event because now we have an enzyme that its only job is to make ATP. The enzyme is called ATPase, and we're going to see these later on. This happens during the electron transport chain. The enzyme is embedded in a membrane. So you can see here phospholipids. So in the case of prokaryotic organisms, this will be the cell membrane. In the case of eukaryotic organisms, this membrane would be the inner membrane of the mitochondria. So remember, to make ATP, we need energy. So the question here is, where is the energy coming from to force a phosphate onto an ADP to make ATP? Well, in this case, the energy is coming from hydrogen ions. Yeah, let me just send a message to the person who cannot hear. Okay. All right. All right. Maybe that would help. Okay, so all right, so the energy is coming from hydrogen ions. Notice that on one side of the membrane there are tons of hydrogen ions. On the other hand, on the other side of the membrane there are very few. So by diffusion, the hydrogen ions want to go to the other side. They cannot go through a lipid membrane because lipid membranes repel anything. The lipids repel anything that has a charge on them. However, the enzyme ATPase allows for the transport of hydrogen ions, and as hydrogen ions go through the enzyme, they make possible the coming together of the ADP and the phosphorus to make ATP. Okay, so oxidative phosphorylation uses an enzyme whose only job is to make ATP by using protons as the energy source to fuel the enzyme. And actually, the hydrogen ions make the enzyme rotate, physically rotate, and bring together ADP and phosphorus. All right, so we're going to go back to oxidative phosphorylation later on. But at this point, you should definitely know the let's see, you should know the two phosphorylation mechanisms of non-photosynthetic organisms: substrate level phosphorylation and oxidative phosphorylation. Okay, now let me go back here really quick. Oxidative phosphorylation. One thing I did not mention about oxidative phosphorylation is that it is incredibly efficient. So this is a very efficient process compared to substrate level phosphorylation. So in the sense that it can make tons of ATP. However, it does need hydrogen ions. So the question is, where are these guys coming from? Where are the hydrogen ions coming from? All right, well, think about the kinds of things that most living organisms consume. Most living organisms, and you can put humans in that group, consume molecules that are rich in hydrogens. We eat carbohydrates, we eat proteins, with lipids. All of these molecules are loaded with hydrogens. So what we would need if we want to make energy efficiently by oxidative phosphorylation is we would need a way to rob the hydrogens out of these compounds that we eat. Well, that process of removing hydrogen ions from a molecule is called oxidation. So oxidation, you probably learn in chemistry, is the removal of an electron from a molecule. And the opposite to oxidation, reduction, is the gaining of electrons. In biology, however, when you think of oxidation, so this is oxidation, these electrons are going to be removed, so molecule A is going to be oxidized. Electrons are given to molecule B, which is reduced. So in biology, oxidation and reduction happens with the removal or the addition of hydrogen ions. A hydrogen molecule has one electron and one proton. So if you remove a hydrogen atom from a molecule, you're oxidizing the molecule. So dehydrogenation is oxidation in biology. This is how biological organisms oxidize, is they remove hydrogens from molecules. So most biological organisms are going to consume hydrogen-rich molecules, and they're going to dehydrogenate them. They're going to oxidize them to capture the hydrogens, which will be used for oxidative phosphorylation to make ATP. Okay. The opposite will be dehydrogenation is hydrogenation, which is given hydrogens, and that is reduction. So this will be oxidation reduction in a biological system. Remember, if you're going to rob something and give it to somebody else, now you're going to have somebody who has been left oxidized and another molecule that's going to be reduced. So in this case, this molecule here is reduced because it has hydrogen ions. Now we're going to oxidize it, remove the hydrogens, and the molecule that gets the hydrogens is now reduced, and molecule A has been oxidized. The molecule that is reduced was previously oxidized. So this one is reduced, this one is oxidized. At the end of the process, this one is oxidized, this one is reduced. So this is what we call oxidation reduction reactions. In biology, the molecules that oxidize and therefore are reduced are called hydrogen carriers or electron carriers, because these molecules are going to carry those electrons to another part of the cell to be delivered to other molecules. So in this case, molecule B was oxidized, NADH, which was reduced, give it its hydrogen. Now B is reduced, NADH is back to being oxidized, and now it can go back and pick up another hydrogen from A. So these molecules that carry oxygens, in this case from A to B, not oxygen, but hydrogens, from A to B, are electron carriers. The most common electron carrier in living systems is NADH, NAD plus molecule. So NAD plus is the oxidized form, NADH is the reduced form. All right. So nicotinamide adenine dinucleotide is NAD plus NADH. So sometimes I describe it as empty trucks versus full trucks carrying hydrogens from one part of the cell to the next. Other electron carriers that will be seen in this chapter is FAD, FADH2. This is the oxidized form. This is the reduced form. During catabolism of glucose, the molecule is repeatedly oxidized. Which atom is removed from the glucose when it is oxidized? So remember back on glycolysis, and actually I would say not just glycolysis, and it's definitely not just glycolysis. Glycolysis, intermediate step, Krebs cycle. When you took the glucose molecule through all those 3 steps, at the end, this is what the glucose molecule became, just CO2. What do we have to remove out of the glucose molecule to create CO2? Because that would have been that. Maybe, maybe not. Okay. We're going to end up with 6 CO2s though, but anyway. All right. So we have to remove the hydrogen atoms. Now, in order for cells to make ATP and to make any type of catabolic or anabolic reaction, they're going to need enzymes. So enzymes are made of proteins. They are molecules that, by definition, what they do is they speed up reactions. They're catalysts. Enzymes are catalysts. A catalyst speeds up a reaction. And this is essentially what depicts the actual role of enzymes. Enzymes are going to lower the activation energy of a reaction. So if you have a certain reactants, in order to get the product from those reactants in a chemical reaction, this is how much energy we're going to need without enzymes. With enzymes, the energy needed is lowered, is reduced right here, so that the reaction is likely to happen. And this is essentially what the problem is. With biological reactions, we're dealing with very stable chemical components. Think about, I don't know, glucose or an egg, for that matter. That's a biological system. Nothing is going to happen to it. It's not going to explode. It's not going to burst in flames. These molecules that make up living things are very stable. They don't react as easily or rapidly. Okay, so in order to make something that stable react, you have to do one of two things. You have to increase the temperature and heat up the component, or increase the pressure and force molecules close together in order to make them react. Okay, in a biological system, if you put heat or pressure, you kill the biological system, so you can use neither. So enzymes are going to lower the activation energy by providing a room, by providing a place that physically brings these reactants together in a close quarter in the right configuration and forces them to react. Okay, that's essentially how enzymes work and make chemical reactions possible without heat or pressure. You remember this probably from biology, that enzymes are specific for their substrates, so that they only carry out specific chemical reactions. So the reactants will sit in the active site of the enzyme. The reaction will take place, and then the products will leave, and the enzyme now is free to pick up more reactants, more substrate. So they're not used up by the reaction. Because enzymes are specific in a metabolic pathway, each step in the pathway needs a specific enzyme. So just as a review from last semester from 1406, the active site of the enzyme is the place where the substrate fits in, and it's been described as a lock and key mechanism where the active site is specific for the substrate. So we'll have an enzyme substrate complex once the substrate fits inside the active site. Enzymes are made of proteins. Proteins are very sensitive to their environment. So enzymes are sensitive to changes in temperature, pH, the concentration of the substrates, and the presence of inhibitors that could fit into the active site, for example, or bind to the enzyme and change the shape of the active site. So inhibitors will be substances that prevent enzymes from working. So oftentimes enzymes, because they're such an important component, have to be tightly regulated. So cells regulate enzymes by binding to the molecules to the enzyme that allows the enzyme to work or stops the enzyme from working. These regulatory molecules are typically binding to the enzyme in places that are not the active site of the enzyme. So these are what we call allosteric places. So an allosteric activator will be a molecule that binds to the enzyme at a place other than the active site. And when it does so, the active site changes its shape, and now it has the correct shape for the substrate to fit in. So the allosteric molecule makes the enzyme work. So this is an allosteric activator. On the other hand, an allosteric inhibitor is one that binds to the enzyme and changes the active site now so that the substrate now cannot fit in. So before the allosteric inhibitor was put in place, the enzyme was working. Once the allosteric inhibitor goes into the enzyme stops working. Okay. So enzymes, a chemical pathway can be controlled by controlling the enzymes in the chemical pathway. So enzyme inhibitors can be the allosteric inhibitor we just talked about. So an enzyme inhibitor could be of an allosteric type, an inhibitor that binds not to the active site but to some other place, and that's called a non-competitive inhibitor. Fluoride, for example, is a non-competitive inhibitor, which binds to an allosteric site of pretty much any enzyme. This is a very powerful inhibitor that inhibits lots of enzymes and stops them from working. A competitive inhibitor is more specific. It is going to be a substance that is very similar to the substrate and binds to the active site of the enzyme, effectively competing with the substrate for the active site. So this is a non-competitive inhibitor like fluoride, binding to the enzyme. The binding of the inhibitor onto the enzyme changes the shape of the active site, and now the substrate cannot fit in and the enzyme cannot work. So fluoride does that. A competitive inhibitor is one that looks a little bit like the substrate. You can see here, similar enough to where the competitive inhibitor could fit into the active site and effectively block the actual substrate from fitting in. So one thing a cell could do if faced with a competitive inhibitor is to secrete more substrate. So if it makes more substrate, now the chances of the actual substrate reaching the enzyme are higher than the non-competitive inhibitor reaching the enzyme. So one good example of a competitive inhibitor, which is used in microbiology as an antimicrobial drug, are sulfa drugs. So sulfa drug is a competitive inhibitor for bacteria because bacteria can make their own folic acid. And in the folic acid pathway, one of the last steps in the pathway relies on a reactant called paraminobenzoic acid. And paraminobenzoic acid is going to bind to an enzyme, and that enzyme is going to change paraminobenzoic acid into folate. Sulfa drugs have a very similar shape as PABA, so that the drug could potentially go into the active site of the enzyme. However, that will block the enzyme from making folic acid. This is a very good selective toxicity drug because humans do not have this pathway. Humans cannot make folic acid, so we don't have the enzyme that binds to PABA. So the sulfa drug is not going to hurt us. It will, however, hurt the bacterial cell and it will prevent it from making folic acid. Folic acid is needed for the synthesis of DNA and RNA, for that matter. So the cell will stop dividing and die. So we have examples of a non-competitive inhibitor in fluoride, an example of competitive inhibitor in sulfa drugs. Okay. Any questions? So we're ready to move on to actual metabolism. And there are three central metabolic pathways that we're going to look at. There's actually four that the book mentions, the pentose phosphate pathway, but we're not looking at the pentose phosphate pathway. So we're just going to concentrate on three. Those three pathways are glycolysis, the intermediate step, and the tricarboxylic acid cycle, which is also called the Krebs cycle. These pathways can accomplish several things. They can make substrate ATP, a little bit of ATP by substrate level phosphorylation. More importantly, they can oxidize reduced molecules and give the hydrogens to an electron carrier, creating NADHs. These pathways also provide precursor metabolites for anabolism. So glycolysis, for example, provides precursor metabolites to make carbohydrates, more complex carbohydrates, like, for example, glycogen. Also provides the precursor metabolites to make glycerol, to later make fatty acids. Krebs cycle provides the precursors to make amino acids, to make proteins. So these are the anabolic functions of these pathways. We're not going to look at these pathways anabolic functions. We're going to look at these pathways catabolic functions, meaning how are they making ATP and how are they oxidizing and creating and harvesting hydrogen atoms for the oxidative phosphorylation steps. But the point here is that these pathways could be anabolic also. They don't have to be just catabolic. If they go in one direction, they're catabolic. If they go in the opposite direction, they're anabolic. We are concentrating on chemoheterotrophs. We're not going to talk about chemoautotrophs or photoautotrophs or photoheterotrophs and how they make energy, even though some of these pathways, if not all of them, are used in pretty much all of these organisms. We're going to look at these metabolic pathways from the chemoheterotrophs point of view. All right, so remember that chemoheterotrophs, some of them are strict aerobes, which means they have glycolysis, the tricarboxylic acid cycle, and an aerobic electron transport chain. Other chemoheterotrophs may be strict anaerobes. Okay, if they're strict anaerobes, now they cannot have an aerobic electron transport chain. They're going to have an anaerobic electron transport chain. Other organisms are fermenters. Fermenters are going to use glycolysis. They're not going to use TCA to make energy. Instead, they're going to turn to fermentation to make energy. And then yet we have the facultatives, facultative anaerobes, which from chapter 4, we said these guys are the lucky ones because they can do everything. They're going to use fermentation. They're going to act as fermenters. They can act as strict aerobes or as strict anaerobes, depending on oxygen availability. Okay. Let's see. Metabolism. Okay. Yeah, we talked about these already. So the first of these pathways that we're going to look at is glycolysis. You do need to know the places in eukaryotes and prokaryotes where we find the pathway. In the case of glycolysis, it's easy, the cytoplasm. Glycolysis is an ancient pathway. Every single cell has glycolysis. It doesn't matter what its metabolism is. Everybody has glycolysis. It's a very old pathway. The purpose of glycolysis is to take glucose, one molecule of glucose, and oxidize it, which now you know means removing hydrogen ions or hydrogen molecules to produce pyruvate. But notice, so that pyruvate, that glucose is a 6-carbon compound. Pyruvate is a 3-carbon compound. So essentially, we're going to take one molecule of glucose and break it up into two, I don't know what that one is there for, into 2 molecules of pyruvate, 2 3-carbon compound molecules. The pathway has 2 stages, the energy investment stage and the energy releasing stage. In the energy investment state, glucose is going to have to be destabilized because we want to take glucose and split it in half. And glucose is very stable. It's not going to split in half. So now we have to make it less stable. We do that by attaching phosphates to the molecule. But the only way to attach phosphates to the molecule is to get them ATP. So now we need to invest energy by spending ATPase to take the phosphate and attach it to the glucose. The glucose is going to be changed into a fructose. So fructose can be split in half better than a glucose. So now we change a glucose to a fructose and yet add another phosphate. So now we have this fructose molecule that has been phosphorylated by the action of 2 ATPs. And notice that the molecule could very easily be split in half, and each half will take a phosphate group, which is exactly what happens. The fructose 1,6-biphosphate will be cut in half, and 2 molecules will be the result. One is called G3P, the other one is called DHA. And notice that they can be changed one into the other. So now we have 2 3-carbon compounds. Well, first we have 1 carbon compound. In the process, we have spent 2 ATPs. Okay, then comes, again, this is the DHA and G3P, the hydroxyacetone and glyceraldehyde 3-phosphate. The next step is to make sure that the DHA is changed to G3P. So now we have just G3Ps, which are 3-carbon molecules with a phosphate group attached to them. So now we're going to oxidize this molecule. We have 2 of these molecules. We're going to oxidize each twice. So now we have 1 NADH for each G3P molecule being produced. We are going to remove the phosphate group and give it to an ADP to make ATP here and here. So now we have 2 ATPs that we have made. We proceed to add another phosphate and remove it. Okay, so another phosphorylation step towards the end to give us 2 3-carbon pyruvates. So 1 oxidation step per G3P, which means 2 oxidation steps. So we're making 2 NADHs, and we're going to make 2 ATPs per G3P, which means 4 ATPs. Okay, so this is just showing you the 2 ATPs being made and 1 oxidation step. Yeah, the ATPs are being made by substrate level phosphorylation. It's just the removal of a phosphate from one molecule and giving it to another one. So definitely know what are the end results, the end products of the energy generation step. What are we producing? 4 molecules of ATP, 2 molecules of NADHs. Now, we did produce 4 molecules of ATP that have been generated by substrate level phosphorylation. However, we spent 2 molecules of glucose, 2 ATP molecules at the beginning. So the net gain is 2 ATP molecules. 4 minus 2, the 2 is spent, is a net gain of 2. So at the end of glycolysis, per glucose molecule, we have 2 ATPs, 2 NADHs, and 2 pyruvic acids. Okay. So in a eukaryotic organism, the pyruvic acid will be moved inside the mitochondria. And the enzyme that does that is oxygen-sensitive enzyme. So that if there is no oxygen, the enzyme will not transport pyruvic acid into the mitochondria. In the case of prokaryotic organisms, there is no mitochondria. So the next step, which is the transition step or intermediate step, happens also in the cytoplasm. So you need to know the location of the intermediate or transition step in both eukaryotes and prokaryotes. What this step is going to do is it's going to take pyruvic acid and is going to oxidize it, decarboxylate it, and destabilize it, make it into a high energy molecule to enter the Krebs cycle. So this is showing you the matrix of the mitochondria, and here is the pyruvate coming in, and this is the intermediate step, converting pyruvate to acetyl coenzyme A. So this step is actually very simple. Here's pyruvate, 3-carbon compound. Remember, there's going to be 2 of these that were made in glycolysis. So this pathway will happen twice per glucose molecule. The pyruvate is decarboxylated, so CO2 is removed, meaning that from a 3-carbon compound, now we have a 2-carbon compound called acetate. Acetate is then oxidized, a hydrogen is removed, making NADH. A high energy molecule called coenzyme A is added, and now we have acetyl coenzyme A, which is a high energy molecule. So at the end of the intermediate step, per glucose molecule, we would have 2 CO2s, 2 acetyl coenzyme A's, 2 NADHs. So we started out with a 6-carbon compound. Now we're down to 2 2-carbon compounds. We've lost 2 carbons in the form of CO2. Okay, moving on to the Krebs cycle or the tricarboxylic acid cycle. Again, location for eukaryotes, matrix of mitochondria. In prokaryotes is the cytoplasm. Now, what we have now is a 2-carbon compound called acetyl coenzyme A, which still has hydrogens in there that could be removed. So we could oxidize it further, which is what the process of the TCA cycle is. It's going to oxidize the acetyl coenzyme A further, getting more hydrogen atoms out of it. So here's the acetyl coenzyme A, which enters the cycle by joining a 4-carbon compound called oxaloacetate. So a 2-carbon compound joins a 4-carbon compound, and we end up with citrate, which is a 6-carbon compound. Now these molecules will begin to be oxidized. We have 2 oxidation steps, here and here. We have 2 decarboxylation steps here and here. And now we are back to a 4-carbon compound, which is one of the compounds we started with. The acetyl coenzyme A has been eliminated through CO2s. The 3-carbon compound is used in a substrate level phosphorylation mechanism to make ATP. The compound is oxidized twice more before we're back to having oxaloacetate. So the tricarboxylic acid cycle is going to give us 1, 2, 3 oxidation stages with NADH, 1 with FADH2. So 4 oxidation steps. We're going to make 1 little ATP. We're going to decarboxylate twice. And because we have 2 acetyl coenzyme A's, we're going to go through this cycle twice per glucose molecule. So per glucose molecule, we're going to get 6 NADHs, 2 FADH2s, 2 ATPs, 4 CO2s. Yeah, remember the molecules of ATP are being made by substrate level phosphorylation. The Krebs cycle also gives us tons of precursor metabolites. These molecules here, the cell can take to make amino acids. So the Krebs cycle is one of the mechanisms through which cells make amino acids. So even if the cell is not using a Krebs cycle to make ATP, it will still have a Krebs cycle because it needs it to get the 5 carbon compounds needed to make amino acids. Okay, so far what we have seen is glycolysis, intermediate step, and Krebs cycle, in which a little bit of ATP has been made by substrate level phosphorylation, but more importantly, lots of NADHs and a little bit of FADH2s collecting hydrogen atoms have been gathered. So the most important aspect of all these reactions when it comes to making energy is that we have lots of oxidation going on. We have lots of electron carriers ready to give those hydrogens to the electron transport chain. That's the key. When it comes to energy making and the energy production, the purpose of glycolysis, intermediate step, and Krebs cycle is to rob hydrogen atoms to make, later on, to make ATP. So the electron transport chain occurs in a membrane. In eukaryotes, it happens in the inner membrane of the mitochondria. In prokaryotes, in the cell membrane, because we need a membrane for the electron transport chain. So this is showing you the location of the inner mitochondrial membrane in eukaryotes. All right, so this is the electron transport chain of a eukaryote, because you can see here that this is a mitochondria. This is the outer mitochondrial membrane. This is the inner mitochondrial membrane. Within this mitochondrial membrane, we have what we call electron transport molecules, which are able to rob electrons from NADH or FADH2s and pass on those electrons between one and the other. Now, a hydrogen atom is made of a proton and an electron. So what's going to happen here is that the electron will be robbed to be shuttled between these molecules. The hydrogen atom, on the other hand, or ion, I should say, the hydrogen ion, the proton, will be pumped against the concentration gradient into the other side of the membrane. So this side of the membrane has lots of hydrogen ions. And in order to pump more hydrogen ions to that environment, you need energy, because this is going against the concentration gradient. So we're going to need energy to do that. Well, electrons are energy. Electrons are high energy particles. So the electron from these hydrogen atoms that were gathered by NADH are going to be passed on from one electron carrier to the next, to the next, to the next, like a hot potato. And the energy of the electron is going to be used to pump the hydrogen atoms to the other side, hydrogen ions, sorry, to the other side of the membrane, building up this concentration gradient, hydrogen concentration gradient. Okay, so you can see here how these hydrogen ions are being pumped as these electrons are being passed on like a hot potato from one carrier to the next, to the next, to the next. Now, this is the problem. You have this hot potato being passed on from one carrier to the next, to the next, to the next. What are you going to do with that hot potato at the end? You have to give it to a molecule capable of accepting these electron, which now is at a lower energy level because some of its energy has been used for the transferring of the hydrogen ions to the other side of the membrane. So we need a molecule, an atom that is a really good oxidizer. We need an atom that can take electrons even if they don't have as much energy anymore. That molecule is oxygen. So oxygen is the final electron acceptor, which will pick up the electron, pick up some few hydrogen atoms from the environment, and turn to water. So that's the final fate of the electron, is to be given to the oxygen. The oxygen will pick it up, and it will be turned into water. Now, this has a couple of problems here that we'll talk about in just a second. Now, the hydrogen ions that have been pumped to one side of the membrane now are ready to come back through the ATPase, which is also called an ATP synthase, to fuel the enzyme which is making ATP. So while the hydrogen ion goes through it, ADP and phosphorus are picked up and ATP is being made. Now, this electron transport chain shown here is an aerobic electron transport chain because it's using oxygen as the final electron acceptor. This is the type of electron transport chain that will be seen in a strict aerobe or a facultative anaerobe or a macroaerophile. So strict aerobe, facultative anaerobe, macroaerophiles are the organisms, the type of bacteria that have these electron transport chain, this aerobic electron transport chain. Part of the problem with the oxygen is that, yes, it can make water, sorry. However, sometimes it also goes around oxidizing other molecules. It can make oxygen radicals, which are poisonous molecules, for which we need catalases and superoxide dismutases. So the four types of electron carriers embedded in the electron transport chain, these guys right here in blue, those are molecules like flavoproteins, iron sulfur proteins, quinones, cytochromes, okay? And this is a electron transport chain from a eukaryote. And so complex 1 is the first one that accepts the hydrogen ion and the electron from NADH, gives it to coenzyme Q, which shuttles it to complex 3. And notice that complex 1 and complex 2 pump hydrogen ions to the other side of the membrane. Cytochrome C shuttles the electron from complex 3 to complex 4. Coenzyme Q makes up complex 2. Again, complex 4 shuts or moves hydrogen ions to the other side. This is the last of the complexes. The electron is now given to oxygen. The FADH2 comes in this area. Gives is hydrogen to complex 3. Okay, so more of the same. Since these are not oxygen, are they like antioxidants? Are you talking about the electron carriers? This is an aerobic. Yeah, the electron carriers, no, they're not antioxidants. They're just electron carriers. However, the quinones and cytochromes, no, they're not antioxidants. However, the antioxidants are going to be the molecules that are going to prevent or destroy the oxygen radicals being made by the oxygen. So quinones and cytochromes are just electron carriers. And they pump the hydrogen. Yeah, when you talk about antioxidants, those are the molecules that will destroy oxygen radicals being made by oxygen. Okay, so just here's the ATPase making the hydrogen, the ATP. Okay. All right. Anaerobic respiration. So we talked about an aerobic electron transport chain, which means there is such thing as an anaerobic electron transport chain. So an anaerobic electron transport chain is one that uses something else other than oxygen as the final electron acceptor. The something else can be nitrate, it can be sulfur. So over here, instead of having an oxygen molecule, there may be a nitrate molecule that gets the electron and turns into nitrite, for example. So that would be the final electron acceptor. So examples of other final electron acceptors, the ones used by anaerobic electron transport chains, will be nitrate, sulfates will also be it. Okay. So this is what is strict anaerobes may do. The electron transport chain generates the proton motive force, meaning the protons which drive the ATPase. So what is fueling the enzyme that makes ATP are the hydrogen ions gathered from NADH and FADH2. For every FADH, there is production of 3 ATPs. For every FADH2, there is production of 2 ATPs. And this is an actual picture of the ATPase. No, nitrogen does not make as many radicals as bad as oxygen because it's not as good an oxidizer as oxygen is. So it is not as poisonous as oxygen is. So yeah, none of these anaerobic respiration organisms that use these other molecules are going to be damaged as badly as those of us that use oxygen. However, the caveat is that because they're not as good as oxygen, they also are not going to be able to produce or cause the production of as much ATP. So an anaerobic respiration pathway is less efficient. There's less ATP made. Basically because there will be less hydrogen ions being pumped because they're going to have to pick up, instead of going all the way to the, let's say, to complex 4, the electron is going to have to be picked up sooner. You cannot let it lose much more energy, otherwise the electron acceptor is not going to accept it. Oxygen is good at accepting low energy electrons. Nitrogen, sulfur are not as good, so they cannot go further into using that electron. It has to be removed sooner, which means less hydrogens are being pumped to the other side. So it's a less efficient electron transport chain, but is a less poisonous electron transport chain. So you cannot have it all. You cannot have efficiency and be protected from poisonous oxygen. You have one or the other. Okay. So essentially what is moving the ATPase, the energy provided to make ATP, is hydrogen ions. Okay, so when all is said and done, typically when we go through all these, we calculate how much ATP is being made. Nowadays, we really need to make sure that the students understand this is all theoretical, that in real life there will be too many other factors that will prevent 100% efficiency of a system. So from glycolysis, we ended up with, actually, let me go this way. We make 2 net ATPs in glycolysis. We also gathered 2 NADHs, and remember, each NADH gives us 3 ATPs, so that will be 6 ATPs made from the NADHs of glycolysis. These ATPs are made by oxidative phosphorylation, versus these 2 ATPs were made by substrate level phosphorylation. Transition steps gives us no ATPs, however, it gives us 2 NADHs, from which 6 ATPs were made by oxidative phosphorylation. The tricarboxylic acid cycle gives us 1 ATP, or 2 per glucose molecule, by substrate level phosphorylation. A total of 6 NADHs, which gives us 18 ATPs, 2 FADHs, which gives us 2 each, that will mean 4 ATPs per glucose molecule. So when you add it up, 34 ATPs are made by oxidative phosphorylation, and 4 ATPs are made by substrate level phosphorylation, for a total of 38 ATPs per glucose molecule. Okay, so you should know where all those numbers come from. Okay, so we talked about everything before ETC is substrate phosphorylation. Yes, exactly. Everything before ETC is substrate level phosphorylation. Absolutely, exactly. Yes. The only mechanism that gives us oxidative phosphorylation is the ETC. Yep, that's a good way to put it. Exactly. Okay, so let's talk about fermentation. Okay, let me show you the definition of fermentation. It is a process that uses pyruvate or a derivative of pyruvate as the terminal electron acceptor for NADH, which is a mouthful and it might mean nothing to you. I'll show you what that means here in just a second. Do know the location, and the location is exactly the same as that of glycolysis. So cytoplasm of eukaryotes and prokaryotes. Again, this is an old ancient pathway. You can argue before organelles came about. So it's all in the cytoplasm. All right, so let me show you the story of fermentation. This is what happens. You have glycolysis. Let me erase that. Okay. Glycolysis, which takes glucose and makes 2 pyruvates. Then the pyruvates are taken through the intermediate step, which gives us 2 acetyl coenzymase. And these are taken through Krebs cycle, TCA, which is going to produce all of these NADHs, which are then taken to the ETC. Now, in the absence of oxygen, so if there is no oxygen, now, unless you have an anaerobic ETC, you cannot use the ETC. Okay? If you cannot use the ETC, really, there is no need for the TCA? No, fermentation and anaerobic respiration are not the same. For one thing, the minute you mention the word respiration, you should say that is an electron transport chain. Respiration and an electron transport chain are the same thing, in the sense that respiration refers to an ETC process. Fermentation is not ETC at all. It's something completely different. So essentially what this is saying is, in the absence of an ETC, in the absence of a TCA, in the absence of an intermediate step, all of a sudden, none of that is available to a cell, whether because the cell doesn't have the ability to use it or because there's no oxygen. For whatever reason, all that is not available. Now we have a problem because the only source of ATP at that point is the glucose, which is going to be those 2 ATPs per glucose molecule net, and it's going to be making NADHs. Now, normally, those NADHs were supposed to go to the ETC and come back as NAD+. But now, the electron transport chain is gone. It's not being used, or it's never, or it's non-existent. Okay? So, at that point is when fermentation is the pathway that saves glycolysis, because fermentation is going to use pyruvate to regenerate, to oxidize NADH back to NAD+, so that it can go back and keep glycolysis going. So fermentation is a pathway that is needed when there is no ETC. And that's probably the best way to put it. Whether it is because the organism has no ETC or because the organism cannot use the ETC. Whichever the reason, the ETC is gone. If the ETC is gone, the pathway that regenerated NADH and oxidized it back to NAD+ is gone. So glycolysis is now going to stop. Fermentation to the rescue. Fermentation is going to take NADH and turn it back to NAD+, so that glycolysis does not stop. That's why it's defined as the pathway that uses pyruvate or a derivative as a terminal electron acceptor for NADH. So, what kinds of organisms are going to ferment? Okay, for one thing, when you think of fermentation, think of an aerotolerant. Aerotolerants have no ETC. So they have no choice but to ferment. Think of a facultative anaerobe. If there is no oxygen, now they're not going to use their ETC, they're going to turn to fermentation. Think of a strict anaerobe. Some strict anaerobes have no ETCs. So they have no choice but to ferment. Okay? Or maybe they don't have the nitrogen that they need for their anaerobic ETC, in which case they have to turn to fermentation. So if the organism has no ETC, like the aerotolerants, or cannot use their ETC for whatever reason, they're going to turn to fermentation. Because at that point, the only pathway that is going to give them ATP is going to be glycolysis. So they need to keep glycolysis going. Now, notice something, that what I said is no ETC, no ETC, no ETC. I didn't say anything about the Krebs cycle or the intermediate step. Those two may still be going on, not to make ATP, but because they're important in making other things like proteins, like the precursors to amino acids, not proteins, but the precursors to amino acids or fatty acids for that matter too. So they may still be going on, but not with the purpose of making ATC. They'll be going on for other purposes, okay? But the key to fermentation, the reason why fermentation needs to happen is because there is no ETC or the ETC cannot be working, cannot be made to work. Okay, so problem with fermentation is that there are many different types. we're almost there. Say there's almost infinite types of fermentation. In general, there are many end products, and we name fermentation pathways by their end products. Okay, so in general, bacterial fermentation yields acids, yeast fermentation yields alcohols. So this is why I tell students, you get grape juice and you put bacteria in the grape juice, you're going to get vinegar. You get the same grape juice and you put yeast in the grape juice, you're going to get wine. Okay? Because in general, bacteria will make acids when they ferment, and yeast will make alcohols. These are some of the products of fermentation. What do you think ethanol comes from? There you go. So fermenters don't use TCA as a catabolic pathway if that was on a test. Yes, correct. They're going to be using it as an anabolic pathway to make amino acids. Yep, that's exactly right. They still have it. Everybody has a TCA because it is needed to make amino acids. So whether it is being used to make ATP or to make amino acids, everybody needs it. All cells need a TCA. So absolutely, yes, exactly. Good point. This is one of the most simple forms of fermentation. Glucose, okay, this is glycolysis right here. Pyruvate is being made. Fermentation will take lactic acid fermentation, will take pyruvate and essentially give it a hydrogen from the NADH, because now NADH will be converted to NADH2, to NAD+, and the pyruvate that is reduced now to lactic acid, okay, when it receives the hydrogens. So this kind of fermentation will be called lactic acid fermentation, because we call fermentation by the name of the end product. This one is ethanol fermentation. In this case, pyruvate is decarboxylated to make acetaldehyde, which in turn is reduced with the hydrogens of NADH to make ethanol. So this is ethanol fermentation. And this is just showing you different types of fermentation. E. coli, when they ferment, they make a mixture of acetic acid, lactic acid, succinic acid, ethyl alcohol, so a little bit of alcohol, CO2, and H2O. We call these mixed acid fermentation, since it has so many acid products. Another bacteria called Enterobacter, when they ferment, they make formic acid, ethyl alcohol, lactic acid, butyriol, CO2, and H2. Because there's alcohol being produced, butyriol, well, there are acids also, but here's another alcohol too. We call these butyriol fermentation. Yes, they make all of those at the same time. Absolutely. Yeah. So the yes, by the same species. Absolutely. Yes. Yeah. So one of the ways to identify Enterobacter is to test for the products of fermentation. One of the ways to differentiate E. coli from Enterobacter is to test for the products of fermentation. Yeah, we do that in the lab all the time. So because some of these species are so specific as to what they produce, we can test for the products of fermentation, and that helps towards the identification of bacteria. Yeah, absolutely. And so this Clostridium is an anaerobe, strict anaerobe, and it's making butyric acid, alcohol, acetone, isopropyl alcohol. So if you ever thought about where does isopropyl alcohol come from, right here. So the factories that make isopropyl alcohol, they have bacteria growing in continuous cultures, making them ferment, and then they isolate the isopropyl alcohol from the other products of fermentation. Ever ask yourself, what did, well, vinegar, right there, acetic acid come from? A Propionobacterium, that's one of the bacteria. There are others that make acetic acid, but that's one of the ones that make acetic acid. Oh, you ever ask yourself, where does nail polish remover come from? Right there, product of fermentation of Clostridium. Okay. So yes, they're very specific as to what they make. I imagine you can tweak the kinds of sugars you give the organism, and you may get some products more than others. Maybe the strain of the bacteria may give you more of one product versus another. So industrially speaking, they probably know how to make the bacteria give you what they want. Is ethyl alcohol made as a competitor during death phase? Okay, it could be during the later stages. That would be in the log phase more so than the death phase. At the end of the log phase, yeah, that could be absolutely. And so yeah, so the commercial producers will have to keep the culture at this specific phase when they're producing these products. Yeah. So we definitely use the end products of fermentation to identify organisms. So we have a test called the methyl red VP test. Actually, it's two tests, not one. The methyl red test and the VP test, which tests the methyl red test for mixed acids and the VP test for butanediol. So that if you have a bacteria and you don't know whether you have E. coli or Enterobacter, you do a MR test and a VP test on the bacteria, and if it gives you MR positive, VP negative, you can say, well, I think I have E. coli. If you get an MR negative, VP positive, you can say, oh, I think I have Enterobacter. So it is one useful way to try to identify bacteria. Okay. All right. So strict aerobes are the boring ones. All they can do is aerobic fermentation. That's, by the way, also us. That's pretty much all we can do, except for our skeletal muscle cells, which can go lactic acid fermentation. But for the most part, all our cells just go aerobic respiration. Strict anaerobes will either go anaerobic respiration or fermentation, depending on the conditions. Facultative anaerobes, they can do it all. They can do aerobic respiration, anaerobic respiration, and fermentation. Aerotolerance, fermentation only. They're the unlucky ones. And notice that fermentation only means very little ATP being made, meaning poor growers. They don't grow very well at all. Okay. Questions? There is a chart. Yeah, I know. So many. Oh dear. Okay. There is a chart for chapter 6 that you want to access and take a look at, in which summarizes all these information. I do have another class to go to, unfortunately. We're going to meet again on tomorrow, lab 6. Yeah, that's tomorrow. Yeah, so let's if you have any questions for this chapter between now and tomorrow, bring them tomorrow. We can address them. I have no problem doing that. Okay. The chart helps putting it all together, and a lot of what you're going to be asked will be, just the basic information from the charts. What pathway takes place? How many oxidation steps? Is there any ATP being made? Is there any are there any decarboxylation steps? What's the end product? Okay. So, all right, I am going to have to go. I will post these on YouTube so you guys can have access probably by tomorrow morning, because this is a long recording.